brings us to Jeff Galvin, who's the CEO and co-founder of American Gene Technologies. Jeff was a serial entrepreneur out in Silicon Valley, founded a number of technology companies, sold them, either went public or sold them to public companies. And one of the things he did was to see that, gee, if you can fix defective or malicious code in a computer system by sending out updates and fixing all instances of that code, why can't you do the same thing in a biological environment? So he founded in 2008, I believe, founded American Gene Technology to do exactly that, working with delta gamma T cells and gamma delta T cells, and he's found a way to actually, and he'll tell you more about it, find a way to cure HIV and a number of epithelial cancers. So Jeff, take it away.
Great, thank you, Mark. For those of you standing, feel free to grab a seat if you want. We're going to chat for about maybe 25, half an hour, and then open it up for questions. But one thing that I was very struck with by Jeff when I first met him was his boldness, basically. I'm going to cure AIDS and I'm going to cure cancer. So I want to get into that, but before we get there, can you give us a little background on, you graduated from Harvard, and then just get us to the point where you sold your first company.
Sure. So I was a computer nerd in the 7th grade, which is before most of you were born, I'm sure. And so about 45 years ago, almost 50 years ago. And I ended up teaching at MIT in my teens on weekends because I was just so excited about computers. And the reason that I got so excited about computers was that my first exposure to them, I got a sense for the immense amount of things that they could do that would create value. And one of my passions in life is reducing suffering for human beings. And I think of all the rote activities that people are forced to do, manufacturing stuff or even driving their own cars instead of having the cars drive themselves, as being things that take away from the more creative and fun aspects of life. And so I think I was really attracted to technology in terms of, how it impacts the public. So I ended up just coincidentally wandering around MIT, and I got a chance to see things like sensor technology and robotics. And so it was the future of computers were clear, and I was totally hooked, and I got grabbed to teach a weekend class there. I ended up being admitted to Harvard, and I was a undergraduate teaching fellow. That's Harvard's snotty way of saying teaching assistant. And I actually managed 30 teaching fellows for the second largest course on campus, which was computers for non-computer scientists. And that was just absolutely perfect for me because what I wanted to do was evangelize my love for computers and to explain to everybody that no matter what career you go into, and remember, I'm talking this was the late 70s. This is mini computer days. The microcomputers were just coming on the scene. And, what I wanted to explain to them is it doesn't matter what you go into. You will either be programming a computer, working with somebody who programs a computer, or working with computers that have been programmed. And all of this knowledge, no matter where you go, will enhance your career in anything that you do. So I was in touch with an industry that was going to impact everybody's lives. So I ended up being recruited in Silicon Valley. I spent time at HP, and then I had a extended stay at Apple, which really just, I think of that as my street MBA.
What year was this around?
That would have been 81 when I went to Apple. Right. Yeah. And Apple really had a vision for computers that matched mine, which is that it can enhance anybody's life. And Steve Jobs had an amazing sense for what would make computers usable and approachable, right? He was the first person to say, hey, put it in a plastic box. This could be like an appliance. It could be in every home. That made it more touchable, more approachable. I mean, that was his marketing genius. But then he also spotted something that I had seen actually a couple years before that, because I had a friend who went to Xerox PARC. And that was the future of computing, the graphic user interface. And you know, by the time I got to Apple, they were already implementing that. I was on the Lisa team, and I just thought, okay, this
Lisa
company
was the
gets it.
precursor to Macintosh, or were they simultaneous?
That's correct. Yeah.
Okay.
As a matter of fact, Steve Jobs was heading the Lisa team until he got kicked off of it because there was some disagreement between him and the board. And but then he ended up heading up the Macintosh, and then as soon as the Macintosh was done, he claimed victory and wiped out the Lisa. So that's a really interesting part of history, and I got to witness the whole thing on the inside. It was an amazing time. But I learned so much about so many things from Apple. So I ended up a bunch of startups, and I looked at my bank account one day, and I didn't need to work anymore, and I'd been killing it myself for about 20 years, and I thought this was always my dream to retire early. And so I bought a house in Hawaii, and my life was flying back and forth between Silicon Valley and Hawaii. And believe it or not, that gets boring. And and I wanted a little bit more stimulation for my brain. So I decided I'll just dabble in technology again. And that's when I met Roscoe Brady from NIH.
Before you met him,
Yeah.
though, can you talk about Varian? Was Varian one of the companies after you left Apple and you
Yeah,
built
so,
that up?
well, they bought one of the companies that I was a co-founder of, or a principal in. Basically, the work that I did in Silicon Valley started with Apple, and then I got excited about a programmable database because one of the issues with the Macintosh was not enough software. So a programmable database just had this infinite ability to match the computer to specific applications in any field. And it was so easy to put together these applications in the programmable database, beautiful graphic, you know, oriented, you know, things that would hold your hand and make the user interface really easy. And any decent programmer would be able to put together something really useful that would create value and would be easy to sell in any particular vertical market. So I left there and I went over to a company called Blythe Software, and that was a really nice score for me. I made a bunch of money off of that. I was the vice president of sales and marketing, and I took the company from 30,000 a month in sales to 6 million a year, which I'd have to calculate how much of a jump that is, but it was enough to take the company public. And back in those days, that's all you needed, believe it or not. So that was a really nice score for me. Then I went over to a company. I went back to Apple for a while. Then I went to a company, Argus Software, and we actually programmed in Omnis, which was the Blythe Software product, a vertical markets tool for quality assurance of linear accelerators for radiation oncology clinics, which basically it's all of the radiation therapy that you hear of are done on these linear accelerators that are built by companies like Varian, Philips, all the GE. And so what we wrote was software that made it really easy to record all of the quality data and to mine that data for problems before you discovered them accidentally. And that turned out to be really popular, and Varian, who was a big player in this, came along and bought the company, and that was a really nice score too. And then I did some stuff in internet, but mostly where I made money in that was investing in internet companies, and I'm somebody who has a good sense for when the party's over. And so I got out with all my money from the internet, you know, .com boom, and also I was playing the real estate markets, and so I was good at spotting when the party was over there too. So I sold my Hawaii place at a high, and I sold a place in California at a high, and then bought at a low, and you know, so all these things added up, and finally, you know, that put me in my early 40s.
Right, and so up until the point you
Yeah.
met Dr. Roscoe, Roscoe Brady, you were a great trader, great real estate flipper, market timer, and also in tech, right? You had serviced healthcare companies, but you hadn't been in hardcore biotechnology, therapeutics, and such. So what was and you don't see a lot of tech guys do what you do, or I don't at least, where they flip from technology into life sciences, hard life sciences, right? Do you see that very often? You do? Oh, okay. Well, I retract that.
Yeah, he's talking. So I met him before. So he's in data mining of biodata. So yes, there is a
That's all like-
crossover.
Yeah.
Yeah, there's quite a crossover now between computers and biotech, but they are focused more on data-intensive applications.
Informatics and things. I'm talking about
yeah.
what you did, which is
Yeah.
going from tech to actually a therapy company, like a true life sciences biotech
So,
company.
so there's two good reasons for that. If you look at my life, it has been just constantly falling forward into things that I love. You know, what attracts me in is I just fall in love with something. And this has been true about computers. It was true about this biotech venture, and I'll explain a little bit more about how that happened in a minute. But, you know, it even had to do with decisions I made when, you know, I fell in love with a girl in college and dropped out for a year. So I'm a guy who...
He's an all-in kind of guy.
Yeah, all in, absolutely. You should have seen my parents freak out, you know, when I dropped out of
Harvard for
Harvard, yeah, for my girlfriend, and they were like, "Oh my gosh, then she'll get pregnant," and then, you know, so they were very, very nervous about that. It worked out just fine. That was also a learning experience for me. But you know, I went back and focused and got my degree and got out of there and then went on to things that I really love. Because I got to tell you that I'm not a big person for doing all this work in a school and then having to pay them. I love the idea that you work really hard and you learn at the same time, like a place like Apple, and they pay you. That's a much better deal. So what happened was I meet Roscoe Brady, and Roscoe had cured Gaucher's disease. And Gaucher's disease is this deadly metabolic disorder where one broken gene results in you not being able to metabolize lipids in your organs, and they swell and burst, and you die. And there was no cure for it. And what he did was he discovered that the missing enzyme was something called glucocerebrosidase. And then he was able to make it in bioreactors a la the sort of Genentech revolution. Instead of making insulin, he was making this enzyme. And then he pioneered enzyme replacement. And of course, you put the enzyme back in, and the missing thing is now back, and it mitigates the disease.
Take a step back. The enzyme back in where?
Oh, in the body, right?
So,
because you're missing it in your
how?
bloodstream. Yeah, by shots every day, right? I mean, it's terrible, but it's way better than dying, right? It's like, you know, it's like having diabetes or something, right? But you don't even have to measure. All you have to do is take the same shot every single day. So that he decided, okay, there's got to be an easier way to fix this thing. And this was at the beginning of viral vectors. Now, viral vectors are a really simple concept. If you don't understand what they are, let me explain it in a minute. And that is that viruses have been around hijacking our cells for 1.5 billion years. I mean, as long as humans have been on the planet, or even animals in general, there have been viruses hopping around. And what they do is they carry genetic code that when they drop it into the cell doesn't know the genetic code that it just got dropped on it versus the code that's in your DNA. It looks the same, and your cell just gets hijacked and produces whatever that genetic code says. And viruses have the ability to go ahead and infect cells and carry these components. And that was very similar to what Genentech was doing, except for Genentech was making those little code segments, putting them in the food for cells, shocking the cells, which would get them to suck it up. And then they'd start producing things like insulin, right? They'd throw insulin genetic constructs in there, shock the cells, and then they produce the first purified human insulin, which is what got diabetics off of bovine insulin. So, but the viruses are much more elegant because you don't have to do everything ex vivo. You can take a virus and you can infect somebody in the clinic. And if you infect cells with something carrying one of these genetic constructs, when it drops it in there, it hijacks the cell, right? And now, of course, all viruses are hijacking your cell for something malevolent, usually reproducing the virus. But what you can do now, and what viral vectors are all about, is scientists have discovered that you can crack any of these viruses open. You can scoop out their viral DNA or RNA or whatever the genetic construct is and throw it away. Then you can replace that with whatever construct you want. And that viral capsule will go ahead and bring that genetic construct into cells, and you don't have to pull the cells out of the body to do this. You can literally infect people in the clinic, and you can put new genetic information in there that will create what they're missing. So the application of this is obvious, right? There's 7,000 monogenic loss of function disorders, including this one that Dr. Brady had cured for Gaucher's disease. And all that means is that you have one broken gene that's not making an enzyme or a protein that you need, and the disease is associated with that missing enzyme. That's what causes the symptoms. So if you can go ahead and get that gene somewhere, and by the way, the industry has progressed so much that I could pull out my cell phone and order a gene over the internet. It'll show up at my company the next day in a plasmid. We can slice it up and stick it in a viral envelope, and we could start experimenting with it same day. That's how far this industry has come along. So you just get that gene, put it inside a hollowed out virus, which is now like a stealth bomber, right, that can deliver any payload. And so the whole idea of viral vectors is converting viruses into updates. Viruses make you sick. Updates improve the working of your cells. Now, what can you do with that? Well, you could just go ahead and correct a genetic disorder. If you have something like in the liver and you're not producing an enzyme, well, that's a nice small localized area where we could deliver what we call a lentivirus, and we're actually doing this in one of our products, which is hollowed out HIV. How's that for scary? But once you hollow it out, it has no disease in it anymore. It can't give you AIDS. Like I said, it's an empty delivery vehicle. It's like a diskette for the human computer, the human cell. It's got nothing on it. You put the diskette in, it does nothing, but it's the software you put on that diskette that's critical.
Does this work on all viruses?
Yeah, you
As
can
far
pretty
as you
much
can
slice and dice almost any virus as far as I know. Yeah, and remember, I come out of computers, so my vision for this is at 30,000 feet. This is why I refer to, the virus as a diskette, and, and I see a whole software revolution, which is in the writing software to improve the human computer, to make it operate better, writing software for better health. So you can go ahead and you can eliminate these monogenic diseases by putting the gene that you're missing into the right place. We do that for phenylketonuria. We just put a small injection of lentiviral vector into the low oxygen region of the liver through the portal sinus, and it infects the liver with a phenylalanine hydroxylase gene, which we've redesigned to get enough expression. And that gives you enough phenylalanine hydroxylase that you were previously missing in this condition, that the disease just goes away, right? Because you are now normal, right? And it's a one and done treatment, because when we infect you with a lentiviral vector, that gene is in there permanently. That's the beauty of HIV, right? Once that HIV infects a cell, that gene is bolted right onto the DNA, and it will go ahead and express for the life of that cell. So we can permanently correct you, one and done treatments. Phenylketonuria costs $300,000 a year just to make you feel normal, and so you don't swell up and get sick and die, right? And for $75,000 worth of vector into the low oxygen region of your liver, and a very simple procedure, that's it, you're done. You're normal, right? What's the next thing you can do? You can improve cells so that they're better at doing their normal job. Why do you get HIV? It's because HIV virons can infect HIV T cells. The sentinel T cell that was supposed to protect you against HIV is the first cell to get infected because it runs after the infection, and then it becomes the beachhead of the disease instead of the thing that's fighting it. Well, guess what? We can do a small tweak to your HIV T cells. They can no longer be infected. And what do? Same thing that cold T cells do. They clear the pathogen that they were evolved to clear. So your HIV T cells can actually clear you of HIV and leave you permanently immune. We do that in an 11-day ex vivo cell therapy where we pull your T cells out. We go ahead and treat them with a lentivirus that hardens them against HIV entry. So HIV is no longer a factor in terms of modifying them, and put them back into the body in quantities that are equal to memory levels. Like after you clear a virus like the flu, you have a certain number of these HIV or flu-specific T cells. We go ahead and duplicate that condition, and we think we're going into the clinic this year. We already submitted the IND, the final safety document, to the FDA, and we're just resolving 2 questions now. So we think this year we'll go into the clinic and prove that that works. And so that's another thing. So just improving cells, get them to be better at what they're doing, and that Same strategy will work for hepatitis B, HTLV, human papillomavirus, herpes. So we can start clearing these chronic viral infections off of that same platform. And then one of the things that Mark was alluding to is gamma delta T cells. This is, again, a small tweak to your body, which has a big healthcare effect, right? It's writing the minimum software, right? Because coming from the computer industry, don't overcomplicate things, right? The more the software, the more the bugs, right? The simplest change that you can find is likely to have the least problems when you get into the clinic. So we were never in favor of CAR T, which, by the way, is a great product, all right? But all of the problems with CAR T, and I'm guessing some of you don't know what CAR T is, but basically CAR T is they take any T cell out of your body and they modify it so it runs after B cells. And they take your hunter-killer cells and they make them, when they put them back into your body, they run after every B cell in your body, a type of blood cell, and eliminate them. Now, why would you want to do that? Well, all B cell carcinomas, like leukemias, are in the B cells. If you eliminate all the B cells, you eliminate all the cancer, and you can live without your B cells. You just take daily injections of antibodies. Now, that's pretty extreme, right? But if you have an acute case of leukemia, that means you're a certain death in the next 6 months. Now, 60% cure rate. Would you rather take a shot every day or die? You'll take a shot every day. And these things are really cost effective, actually. I don't want to get off on that tangent, but the bottom line is that they're putting people back on the street that are paying taxes and insurance premiums instead of dying at what would have been a higher cost than living now. I mean, it's just amazing, even though these things are expensive. But anyway, CAR Ts have these problems. So what we said is, don't modify the T cells so that they're hypercharged to go up against, a target like a cancer. Just modify the cancer so that it's infinitely attractive to the existing T cells. And that's what our cancer product is. You have these gamma delta T cells, which are 3 to 5% of your circulating T cells. That's constant memory state. They're always busy. And why is that? They are surveilling your entire body for epithelial malignancies. So small occurrences of breast, prostate, lung, liver, colon, kidney, ovarian, pancreatic, head and neck, and skin cancers and other cancers. And they're eliminating those small malignancies so they never turn into solid tumors that are actually a threat. Everybody in this audience has cancer. And it doesn't matter because a healthy immune system will eliminate it before it becomes a health threat. And 2/3 of us will get all the way up to, 90 years old or whenever we pass away, never had an epithelial solid tumor. But when it escapes, right, then these gamma delta T cells can't take care of it anymore. That's when you get surgery, radiation, chemotherapy. Instead, what we do is we locate the primary tumor, no surgery. We put a small amount of viral vector into it. It starts expressing a stimulatory small molecule, which is the thing that attracts the gamma delta T cells. They all come to that one site, that primary tumor. They get trained, proliferated, and activated. In that activated state, they eat at 300 to 600 times their normal rate. They don't just wipe out the primary tumor. They're circulating around your body. They get the secondary tumors, the metastases, and unrelated epithelial solid tumors. That means if you come in with a lung tumor and we treat that, if you had a little bit of breast cancer or a little bit of prostate cancer that hadn't been detected yet, it gets mopped up. It's like resetting your cancer clock, and we never modified anything important or complex like the immune system. We literally modified the thing that's going to be killed by the modification. So it eliminates from your body. This is a really elegant solution, and we have a 1,000-mouse study now of advanced cancers of these types, with 85% of the mice coming back completely cancer-free. You can frozen section the whole body, zero cancer, just gamma delta T cells floating around looking for more victims. So it's very exciting, but this is the future of gene and cell therapy, and this is why I fell in love with it. So Roscoe Brady explains this thing to me, and I've been through the computer and software revolution anymore. I mean, literally at the first meeting, I said to him, somebody's got to be the Microsoft of this new industry, right? And believe it or not, when you come out of biotech, nobody thinks that way. How do you make money in biotech? Find something that works in the mouse, take a $100 million bet, get it into the clinic. The only way you're going to fund that is you got to have brand name executives, brand name technology out of a brand name university and a brand name biotech VC. And, and if you're lucky and it works, which is like at that point, it's probably like 1 in 50 drugs. Yeah, 1 in 1,000 might even be, not unrealistic when you're talking about early stage stuff. But if it works, you're going to be rich. And if it doesn't work, you have nothing. But this isn't like that, right? We're not talking about molecules that can't be changed. We're talking about ACTGs that have very specific behavior when we put them in your cells. It's deterministic. It is really like software. It is debuggable. First of all, you never end up at this situation where you've got nothing. You may have something that has a bug. Did Microsoft pull Excel from the market because it had one bug? No, of course not. You go ahead and you improve it. And in fact, that'll be the future of all these drugs. There'll be V1, V2, V3, V3. You know what I call them engineers, but they're really PhDs, right? I tell them, great, looks like you've solved HIV. Now, solve it better, faster, cheaper, right? Because this is going to be an incredibly competitive industry of software for the human computer. And I think the difference that we have at American Gene Technologies, and I think this is our vision that is unique, and it comes from me coming out of those previous industries, is that there's a pivotal layer in software that allows you to dominate this market. It's the tools and reusable components, sometimes referred to as the operating system, the platform. Platforms are nothing in old biotech. They're everything in this industry. And so what we've done is 12 years of accruing all sorts of critical foundational innovations in the use of viral vectors, any type of viral vector. We call ourselves agnostic. We don't care whether you're using AAV, lentivirus, adenovirus, whatever. We're about the software that goes on the diskette. We couldn't care less who duplicates our diskettes, and we don't care what type of diskettes they are, because we know CDs are coming, then DVDs, then thumb drives, and this is the future of our industry, right? So what we are is we have all these components that you can mix and match to cure a myriad of diseases. What does that make us? The most efficient software developer in the software industry for the next 100 years. And he's nodding because he's a software guy. But if you come from my background, you just instantly realize, of course, how could it be any other way? And we're going to prove that out with HIV. And then that platform could do hepatitis B. We're 80% of the way there on hepatitis B. Try that with a small molecule, right? You find something that cures hepatitis C, you're nowhere on hepatitis B. So this is I fell in love with this stuff when I'm going back to your original question, and I just couldn't help myself. I was like, this is the biggest thing I've ever seen. And Roscoe Brady was retiring, and they were going to throw away his stuff at NIH. I was like, can I have it? They were like, sure. I was like, how much is this going to be? 0. Yeah. So I got all of the most advanced plasmid systems in lentiviral vectors. I hired some postdocs from his lab, and we got experimenting right away. And the day that I knew we had it was Roscoe Brady stayed on as a scientific advisor, which was really important because it gave us cred. It gave us grants that assisted. I put $2 million into this company myself before anybody else joined me in this thing. But also the people that believed in this were NIH, right? Because they've been involved in the Human Genome Project, and Roscoe's was famous at NIH, and he was developing this cutting-edge viral vector technology. So they gave us little grants. We studied 15 different diseases and did 15 different projects and collected all the things that were in common between all of them, right? And that became our platform. So Roscoe explains this stuff to me, and then I find out they're throwing away what I think is the most valuable technology I've ever seen in my life, even more valuable than computers. Remember, computers is $3.5 trillion a year, everything connected to the internet, all those devices, right, including your Amazon speakers and stuff like that. Healthcare, 10.5 trillion. And it's only 10.5 trillion because that's all we can afford to pay in there. But what about if it really made your life better? You might forego that Lamborghini and improve your body instead, because it is actually better to feel good than to look good, right? So, this is an industry that is capped only by the utility that it brings. Look at the $1 trillion pharmaceutical market. I mean, half of those drugs do nothing but give you hope, right? Look at a drug like Avastin, $7 billion a year in sales, 6 weeks of extra overall survival. Think about that. That's all you get from that. Now that's going to be replaced by something that you take at about the same price or maybe 3 times the price, but that will give you 5 or 10 years of knocking back your cancer. That's the future of gene and cell therapy. That's why pharmaceuticals is not a $1 trillion market. It is a market that is $10 trillion that has been held back by the fact that drug developers can't do things that give enough value, right? And we can do that now. This is that powerful technology that will allow us to do it. So, you know, our vision, earlier we had these entrepreneurs up here, or VCs. You want to build out Baltimore? Well, guess what? Once we prove the OS, anybody that knows a disease well can found a company. Baltimore's a great place for it. We're right down there in Rockville. I see Rockville to Baltimore to Frederick as a triangle which is becoming DNA Valley. That is my intent. We'll see if we can do it, right? And I think that maybe what we have in Rockville is the Microsoft of this industry. I don't see anybody else trying to do this. I see everybody doing these siloed developments. Who's one of the biggest players in our industry? Bluebird. They've been in the clinic debugging something that they could have debugged at the bench at hundreds of millions of dollars per month in operating expenses and clinical trial expenses and all that stuff, right? Because they didn't wait long enough and learn the fundamentals of reprogramming the human computer so they could pick an application that, you know, and I really mean it's an app right on top of once you know this stuff, that they could pick an application that would be more likely to work in the clinic, and they could anticipate the stumbling blocks and the landmines before they got there, and it blew up at the most expensive part of the drug development cycle. So I think it's a whole new model. People up in Baltimore will raise $10 to $20 million and they'll cure a monogenic disease that's worth $10 to $20 billion, and they'll start a company, right? Just like the apps on your phone. Nobody will go back, eventually once these things are proven out, nobody's going to go back to writing on the hardware. They're always going to adopt the operating system in between, because on day one, with all that utility, you're 80% of the way there. All you need to know now is what gene you want to express where. And gene and cell therapy not only expresses those genes, but it has targeting capabilities that put the medicine only where it's needed. You can steer the viruses to the right cells, and then you can put these smart switches on the genetic construct that only turn on if they see certain things in the cell that can indicate a tissue type or even a disease state. We can put a human prostate-specific antigen promoter on a genetic construct, and it will only turn on if it's high levels of HPSA. What does that mean? You probably have prostate cancer in that cell, right? So this is amazing stuff, and you're going to be able to do it at a low price. And 7,000 monogenic diseases that now cost $2 to $4 trillion in the healthcare system in palliative treatment, like Gaucher's disease used to be, or still is, right? $300,000 a year. Palliative treatment. They haven't cured anything. They're just treating it for life. There's 7,000 of those, 7,000 of those things that are going to be cured by viral vectors over the next 20 years.
Great. Thank you for your passion and
Going
on like that. We do have time for a couple quick questions. We want to stay on schedule if possible, but does anyone have any burning questions for Jeff? Go ahead. Sure.
Why are you here and not curing norovirus right now?
Yeah, well, there's not a reason why we couldn't make a cure for norovirus, but with gene and cell therapy, you know, these are expensive things. So you go to the most acute things that are the most expensive. And norovirus, you know, can be treated. And so nobody's going to buy a $50,000 cure to norovirus. And so, you know, you have to be practical about it, but we will, and I'll tell you why. Think of the nature of technology, and technology has finally come to drug development. The tool set doubles in capability every year, and the cost halves, right? This was Moore's law in computing, and when you look at this industry, it's actually dropping off much faster in terms of costs. So what does that mean in that environment? It means if you can dream it you can do it. Anything's possible, right? When I saw a teletype, I knew these things were going to be driving our cars one day. We're still not there. I couldn't have predicted when it would happen, but you know it will happen. I'm absolutely certain of that, right? And so that's the nature of this technology too. Remember that nobody even had a phone in Africa, and now they all carry these. Why is that? The cost, boom, the utility, boom, and now it's justified. Even if you're not a rich Western country, you can get these solutions out to them. So we'll cure HIV for the Western world with something that's too expensive for Africa, but it's going to come down in a price range where everybody in Africa is going to get it too. So anyway, so that's why I'm not curing norovirus. The timing's wrong. Or
Yeah.
coronavirus, same thing.
Yes, that gentleman right there.
The HIV one? So cancer, it wouldn't make sense to treat you because, first of all, we need that solid tumor as the site of immunogenicity, right? We need some place to put the treatment. And we wouldn't want to raise the activity of gamma delta T cells for your whole life. We just want to raise them in response to a need. And what we love about this thing is by using the primary tumor, that's the first one that, that one's going to get melted away. It's the biggest one. So while it's melting that away, it's melting away all the other ones that are smaller. And your final timer goes out when your body's clear, right? And then your gamma delta T cells go back to normal. Keeping you normal is what we should be trying to do. We're trying to normalize your body with a gene and cell therapy so you can fight HIV, or you can be cured of a monogenic disease, or you can fight a cancer. But then afterwards, once the cancer's gone, we don't want you different than you were before. And so, that's prophylactically, you wouldn't do that for cancer. For HIV, you could. If anybody that has $500,000 can go down to the doctor once this thing's introduced, and they can be permanently immune to HIV, just like people that are HIV infected. But you might as well wait until you get an HIV positive test. The price doesn't go up. The only difference is you take antiretroviral therapy for a year or two, and then you're eligible for the cure because your HIV T cells are back. So it wouldn't make sense to spend $500,000 for a vaccine on something that is curable, right? So we're doing the cure first, and then we'll work on vaccines though.
So,
Yes, ma'am.
the last two responses have alluded to the cost, and even Bluebird, when you were talking about, they're talking about $2 million for their beta thalassemia treatment, and their plan is to distribute it over 5 years. But what's AGT's thought? Because even in Western societies, that's a big pill to swallow.
Yeah.
Many people can't afford a $500,000 mortgage, much less a therapy on top of all their additional costs.
Okay, so there is hope in this area. One is that just general trend. The prices are going to come down. This is a competitive industry, and there's no way that Bluebird or Avexis, now Novartis, is going to be able to maintain these multi-million-dollar cures when they can be cured much better, faster, cheaper. And we didn't go into those things because we didn't think they were commercial from day one, right? So we went into HIV and we said, okay, what are we spending on HIV right now? Well, it turns out it's somewhere between $80,000 and $120,000 a year per patient. The medicine's only $20,000 to $30,000 a year, right? But the comorbidities of the medication is another $50,000 to $80,000 a year. And so, it centers right around a little less than $100,000 average, which means you get your cost back in 5 years, which means the insurance companies are making a profit after that. Now, you spread it over 5 years, for a $2 million thing, that's still really, really expensive, $400,000 a year. So we think it's possible by choosing the right low-hanging fruit to bring out things that are commercializable and that are practical from a reimbursement standpoint as well. Phenylketonuria, $300,000 a year to treat. Okay, what if it's a $600,000 cure? And it prevents the long-term psychological effects, because even if you treat phenylketonuria, by 20 you're depressed, 30 you're schizophrenic, 40 you're suicidal. It's a slow death, and there's no treatment for that. Would Sigma spend $600,000 to cure you instead of treating you through that cycle? You bet they would. As a matter of fact, they made it official. Sigma's come out and said, we're just going to pay for gene and cell therapies. The head of NICE, which is the national insurance in the UK, you know what he said about CAR T? CAR T is this cancer cure for acute leukemias of the B cells, right, that we talked about earlier. He said, it's a $500,000 product. He said it would be a bargain at twice the price. Now, why did he say that? Because dying ain't free. If you have an acute myeloid leukemia, you don't lie down and expire. You go to the hospital for treatment after treatment after treatment. It costs about $1 million, and then you expire, and then your family still gets stuck with the funeral expense, right? So at $500,000 to put you back on the street. And this is the way we need to think about drug pricing. We need to think about, one, what it's costing now. Can we bring that down? If we can bring it down, it's good for the insurance companies, it's good for society. But then it is, what is the value you get out of each one of these things? When I was at Apple, we introduced the Macintosh at $2,100. Only rich hobbyists could afford to buy one, and it did nothing. But if we were a drug company at that point, and we sold like 20,000 of them the first year, it was a total failure. But if we were a drug company, what would we do? We'd jack the price up to $2,500 the next year with no innovation, right? Because the idea is that if you've got a captive hostage audience and their only hope is your product, you can go ahead and go up 3% per quarter. Nobody's going to notice, and they don't have any choice. You're their only hope. That's not the way it is in competitive industries. This is why that pricing issue is going to disappear on its own. Because if we stick with $500,000 to cure you of HIV, somebody will come along with one for $250,000 that works just as well. You know who that company will be? AGT. Because we haven't forgotten that the most important thing to be is the most efficient software developer. That's why you become Microsoft. How do you become Apple? They have a platform also, but you know what made Apple one of the biggest companies on earth? Different type of platform. They keep you in an ecosystem of hardware, right? Think about the #1 app you use on your Apple AirPad, or probably even your iPad, right? Is Microsoft's products, right? So there's two ways to do this, but platforms are everything. The cost will come down. Why doesn't Microsoft? You have to use Excel. Can anybody get by without Excel and Word these days? So why don't they jack the price up to $3,000? Why is it so cheap? Because there are alternatives, right? So drugs are going to be a whole new thing. Gene and cell therapy will disrupt pharma to the point when, mark my words on this, if you remember nothing from this presentation except for that in 15 years you're going to see companies like Pfizer, BMS, Roche on the bubble. Why do I know that? Because I've seen it already. When we introduced the Macintosh, everybody said the graphic user interface is nothing, but it was really a disruptive, important technology. And the other thing they said is that IBM's going to crush you. Why even bother, right? Where's IBM? They install PCs for a living. They don't even make mainframes anymore. But what I really want to know is where's Burroughs, Sperry Univac, Control Data, the GE mainframe division? Yeah, Digital, these are the mid-tier. They were the first ones to get crushed. Digital, Ken Olsen famously said, he was the head of Digital. This was in the Boston area, #1 entrepreneur. Trust me, you would see him everywhere. His famous quote is, nobody will ever want a computer in their house. I bet you everybody here has at least 5 computers in their house right now. and Apple got told by the #1 analyst that the graphic user interface didn't matter. You know why? Because PC experts like to use their computer by leaning back in their chair, putting their feet on their desk, and putting the keyboard in their lap. So the command line is the most efficient way to talk to the computer. And nobody wants to keep leaning forward and doing the mouse. I mean, think about that. Amazing. Blindness that comes from success, right? And, this is something that as an entrepreneur, you have to remember that you need to trust your vision. And, I always tell people that if I had a nickel for every time an investor or a drug exec or anybody else told me that I was absolutely crazy about this vision, I wouldn't have had to raise money, because that would have been enough money to fund the company right there, right? But, sometimes you actually are onto something. And if you're in love with it, you'll actually have the energy to keep doing it, right? If you're in love with, especially the technology, with your baby, if you really believe in it, you're thinking about it 24/7, and you're going to persevere. And this is going to make it much more likely that you'll make a success out of it.
Great. Thank you so much, Jeff. Let's give it up for Jeff. Thank you.
Thank you. Thank you very much.
Can you stick around, because we have the place thanks to the dean until noon. So if you I'm sure a lot of people have additional questions. And
I'm happy to stick
Oh,
around.
thank you so much.
And
our next CONNECTpreneur is in DC on March 25th. So mark your calendar and hope to see you out there. We have the place till noon. So continue mingling and networking. And thanks for coming out.
Oh, thank you.